Inside every cell of your body lies a blueprint more complex than any building plan ever created. Welcome to the world of DNA sequencing, the revolutionary technology that reads the genetic code within our cells. Today we'll unlock the secrets of how scientists map genomes and discover what it means for medicine and biology. To understand DNA sequencing, we first need to grasp what DNA actually is. DNA consists of 4 chemical building blocks called nucleotides. These bases pair in a specific way, A with T and G with C. The sequence of these bases contains all the instructions needed to build and maintain life. The human genome contains over 3 billion base pairs, and sequencing allows us to read this genetic text letter by letter. DNA sequencing began in the 1970s with Frederick Sanger's groundbreaking method. The Sanger method used special modified nucleotides that stopped DNA synthesis at specific points. Scientists then separated these fragments by size using gel electrophoresis. By reading the pattern of bands, they could determine the DNA sequence. This method powered the Human Genome Project, which took 13 years and cost nearly $3 billion. While revolutionary, Sanger sequencing was slow and expensive for large-scale projects. Everything changed with next-generation sequencing technologies. These machines can sequence millions of DNA fragments simultaneously. The process begins by breaking DNA into small fragments and adding special adapter sequences. These fragments are then amplified to create clusters on a specialized surface. During sequencing, fluorescently labeled nucleotides are added one at a time. Each base emits a different colored light, allowing the machine to identify the sequence. This happens for millions of fragments at once, generating enormous amounts of data. Powerful computers then piece together these short sequences like a massive jigsaw puzzle. DNA sequencing costs have dropped dramatically, fueling research and revolutionizing healthcare. In personalized medicine, doctors can now analyze a patient's tumor genetics. This allows them to choose treatments tailored to each individual's genetic makeup. Pharmacogenomics helps predict how patients will respond to specific medications. For rare diseases, sequencing can provide answers that were impossible to find before. Consumer ancestry testing has made genetic analysis accessible to millions. These tests reveal our ancestral origins and migration patterns throughout history. Large-scale population studies are uncovering the genetic basis of diseases across different ethnic groups. Beyond human health, sequencing is transforming agriculture and conservation biology. Scientists can now sequence entire ecosystems to understand biodiversity and environmental health. Comparative genomics reveals evolutionary relationships and helps us understand life itself. However, this genetic revolution raises important ethical questions. Genetic information doesn't just belong to individuals, it affects entire families. Questions about genetic privacy, discrimination, and data ownership continue to challenge lawmakers. We must balance the tremendous benefits of genetic research with protecting individual privacy. Informed consent and genetic counseling are becoming increasingly important. Ethics committees now guide genetic research to ensure it serves humanity responsibly. The future of DNA sequencing promises even faster, cheaper, and more accessible technologies. Global collaboration is accelerating discoveries that will benefit all of humanity. From treating genetic diseases to understanding human evolution, DNA sequencing continues to unlock life's mysteries. As we decode life's blueprint, we're not just reading our genetic past. We're writing the future of medicine, biology, and our understanding of what makes us human. The genetic revolution has only just begun.